made kits. Yeah, the coronavirus changed the pattern, though. They made kits available. The government paid for those, though, in the United States. So they run. What do they do? Run around $20 now, I think, if you buy them commercially. In that, so it made diagnostics a lot easier for it. So we're entering an age where diagnostics going to be lower cost, simpler to perform. I think with it that we're seeing. That's from the medical standpoint in that. But I don't know if they really do anything in cell culture anymore in the clinical labs. I would be surprised if they did. Used to
across the board, like
pretty much,
across the country, or
I should ask one of my clinical virologists for that. I
did
don't.
that change like when COVID was happening?
Oh, it changes the molecular methods probably right after 2000. It started to change because
hmm,
the molecular methods became the instruments were developed, high volume, automatic. You just put the sample in, it does all the processing in that. So it's gotten a lot. If you have the instrumentation, you can do a lot. A lot of it happened too, but what did COVID did? Wastewater epidemiology came in. Have you talked about that, wastewater-based epidemiology? Well,
no.
okay, it's been used. It was used for polio virus a lot. In other words, when they were doing poliovirus campaigns, and particularly in Africa and Southeast Asia, they learned they could test the sewage and they could detect one case in 10,000 in a community if they found a virus. And if they found a hit for polio, they went in and vaccinated everybody in that community they could. What they learned during COVID, and we use it quite a bit at our university, by testing the sewage, we could tell you how many COVID cases there were in the city and how many there were going to be in the next 10 days. Because we could, you start excreting the virus before you become clinically ill. So what we did with the university, rather than close the university, we monitored all the dormitories every week. And we could tell if there was anybody in that dorm that had a COVID case. And if we got a hit, then when they returned that evening to dorm, they tested the students to see which one was ill. So it became a really useful tool. We could say, well, in the next 4 days, the number of cases are going to increase or decrease. And we could see when they mandated masks wearing on campus, the COVID cases decreased for it. So it was a good indicator of interventions too for doing it. And it was so, I mean, it's done all over the world now. There are thousands of samples collected because they learned they could also look for influenza. They could look for RSV. They could look for norovirus. In other words, you can get a snapshot of how many people are ill in your community from any kind of virus you want to look for, and how many will probably become ill in the next several days. Very sensitive. You can get one person in 10,000 with these techniques in that. Now, the Centers for Disease Control is set up a network of 100 cities to monitor infectious diseases across the country now using the wastewater-based epidemiology. So that's one powerful tool that showed its usefulness in the COVID outbreaks on it, this wastewater-based epidemiology.
Okay,
I don't know if you
so
read the paper, you can see they say the cases are on the increase. That's because they're monitoring the wastewater.
okay. So just going back to the processes that they're using. So you're saying they're not using cell cultures anymore. They're using the PCR tests
Yes,
completely
yeah,
in the labs,
right.
in the virology labs.
Yes, some labs may still use enzyme-linked assays, but yeah, pretty much all molecular now.
Okay, so what usually happens when they have a sample and then they just basically test it?
Yeah, you have
They
to
they
process it
run the
first.
sample.
Yeah, you process it. You have to remove the virus. You have to separate it, and then you have to remove the nucleic acid, and then run a enzyme test on it, which amplifies the nucleic acid. So it does take a little bit of procedure, but some of it's automated now. So you can just
Okay, so how do they remove the virus?
they use an enzyme that chews up the capsid, the coating around the virus, and it
Okay.
expels the nucleic acid then. Or you could use a chemical too, I guess, but that's what we do.
Okay, so it's a chemical that's put into whatever the sample is
Yeah. That's right.
to kind of bring out the thing that you're going to test.
Yes, basically.
Okay.
Instead of the virus. Yeah.
Okay. So how do you know? So you're basically, um, I guess. How does the test know what the virus looks like?
Yeah, what you do is you put in what we call primers, which are will attach to the nucleic acid, and they'll be the catalyst for amplifying the nucleic acid of the virus if it's the virus you're looking for. In other words, you have a different set of primers for norovirus than you do COVID. So if you put a sample in there and there's no amplification of norovirus or COVID, you don't have the virus. Or if there's only,
Okay. Like I understand that part.
yeah.
What I'm saying is, how does the test itself, how does it get programmed to know what the virus is?
It's instrumentation. There's a computer program in the instrument that does it. There's a readout on the screen, and you can see the virus being amplified or not.
Mm, well, no. So I mean, whoever programmed the PCR test
Uh-huh.
had to have the virus to begin with,
Yeah,
right?
right.
So what I'm saying is, how does that person who programmed the PCR test, how do they know that what they're programming into the PCR test is a virus?
Oh, what they're yeah, what it is, it's getting a readout from the replication of the virus. The primers will only amplify a particular type of virus or group of viruses. If it's not amplified, in the sensors pick that up. It goes to the computer, and you get a readout.
Right. I'm talking about before all that happens.
Oh, the
When
primers
the
are
person
designed.
when the person who's programming the PCR test is saying, okay, detect this, how do they come up with that um criteria basically in order to
Yeah.
are you aware of how that process works at all, or
Well, the program, it's a program that tells it that the primers are designed. If you have the knowledge of what the sequences of the amino the nucleic acids of the I'm trying to say the right word. What's the best way to say that? In other words, basically, you're a computer program on a DNA, and it's reading. You are what your DNA reads, and every virus is the
Mhm.
same way, so it's different. So I could tell you're a human. I could tell you come from Europe by looking at your DNA because you have that information in your DNA. Um, uh, and it's the same thing with the virus. We sequenced all the nucleotides. That's the word I was looking for in the virus. In that pattern, and it's a certain pattern. And each virus has its own pattern.
Okay, so
Primers are designed.
who
Uh,
sequences those nucleotides?
who's people? Well, actually, we could do it. We send it to a center, and they sequence it for us when we want to do it. How?
Okay.
I'm not sure exactly how they do the sequencing. I haven't been involved in that. But in other words, they determine the order of all the nucleotides in your DNA or RNA.
Okay. So the way that your practice functions is that you're basically relying on the results of the PCR test to tell you if something is or isn't a virus,
Yeah, or what virus
right?
it is.
Okay.
You have to know what you're looking for.
So what if the sequence that is in the computer to create the PCR test is is faulty? How do you know that that sequence is correct?
Uh, well, you're basing that on the literature of what it is, what it's been published in the scientific literature. Usually
Okay.
more than one, where most scientific work does, like I'll publish a paper on something. Well, you always like to see somebody else repeat it.
Hmm.
So that's one of the way self-checking scientists, somebody will try to repeat it if it's a useful piece of information. So in other words, well, okay, that worked for he's being able to find norovirus. Well, let me try it in my samples of norovirus and see if it works. Uh,
Hmm.
and we
But
do,
all you're doing there is just seeing if the test is going to detect the virus. But you're not determining if the person who submitted the RNA into the computer is actually doing that correctly, if what they're submitting into the computer is actually the RNA of a virus.
yeah, you assume that. And of course, it's probably been published in the literature and this information's available. You can look at it. See what they're using. They don't make it up. In other words, this is based on our analysis of noroviruses and it's been published in the
Uh-huh.
literature. So it can be checked or verified if you want.
Okay, so for neuroviruses, but what about for like COVID virus?
I'm sure it probably has been. Yeah.
Hmm.
I didn't do any, uh, other people in our group do that kind of stuff. But yeah, it's been I mean, that thing's been looked at quite extensively, the coronavirus. Oh, even the variants, when they look for it, that's how they can tell you that all these variants coming around because they're sequencing the nucleic acid all the time.
Hmm. Okay. So, but the issue that I have with um and that the people that I've read
Uh-huh.
about online
Uh-huh.
that they have the issue with is so it is actually pretty crazy to me that you're not using cell cultures, that you're just using the PCR test to determine, and you're saying that most or if not all labs, that's what they're doing these days. Um, but because the main argument that these people make
Mhm.
is that um, the way the cell culture process, because there needs to be some kind of cell culture process or something like that in order to find the virus that can then be uploaded
Oh,
to
yeah.
the
Right.
computer.
Oh, they've done that. They've isolated infectious viruses in cell culture from urine and feces and respiratory secretions. People have published number of papers on that.
Right. So the majority of those papers, if not all of them, require the use of cell culturing.
Yeah, yeah. I mean, we grew up SARS-CoV-2 a lot in our laboratory because we did a lot of work with it for a while. So you can grow up right. Yeah, they've isolated it from clinical specimens, from to see where it was excreted. They know it was excreted in the urine and the feces, saliva, mucus, in cell culture.
Okay.
Yeah.
In the cell culture, but never directly from the sample.
Oh, right. Well, they take the sample of, uh, let's say, mucus and put it in cell culture. That's how you would
Okay.
isolate the
But
virus.
they can't find the virus in the saliva or in the
Oh, yeah,
sample
yeah. They can find it in saliva. Yeah, and mucus, feces. They do find the virus.
without using the cell
Oh,
culture.
they use cell culture. Yeah, they've used cell culture. There are papers published on that.
Right, but without using the cell culture.
Oh, people have done that too. Yeah, that's a typical diagnostic test. Yeah,
Straight from the sample.
right. But for routine clinical work, they don't use cell cultures. Too expensive, time consuming, and the PCR can
Right,
be.
right.
Yeah.
So you're saying the only two things that have been done are the cell culture isolation or a PCR test of the sample directly from the sample.
Right, yeah, from a clinical specimen. Now, people have done
Okay.
work too in hospital rooms showing they can find infectious virus sometimes on surfaces too in the room where people are.
Okay. So minus the PCR test, that has it ever been extracted directly from a sample
Oh,
without having to use the PCR test? You extract it from the sample, you look at it under an electron microscope, and then you find the virus.
oh, I believe people have done that. I can't remember. I've seen EMs of it, so I assume that's the case. It's a possibility.
Okay.
But it's not that easy.
So
You have to realize you have to have a lot of virus before you can see it under EM. It's not that easy.
okay. So if they did it that way, they would still need to prove that it's a virus and not just a random particle in that sample, correct?
Um, well, yeah, viruses look like viruses. I'm not sure what a random particle would look like. You can see the proteins in the capsid in the EM. If you have a good EM, you can see
Okay.
the capsomer. They call them capsomeres, the rounded thing. But you can see them. I haven't looked up photograph for a long time for it, but you can find it. It's a little bit harder with um, coronavirus because they have a lipid layer, but you can tell the difference between a particle and a virus. At least I could, all the work I did.
How about an exosome and a virus?
Yeah, you should be able to do that. Viruses are a lot smaller, and they look like a virus.
Okay.
And you
But
can
so
always
looking,
in other words, are you seeing something anybody? I would have doubts about it. A good person with an electron microscope could do this really easy.
okay. So, but you're basing if it's a virus or not on if it looks like a virus.
Yeah.
Shouldn't it be based on if it actually is a virus, meaning if it creates pathogenicity,
Well, yeah. Well,
which is
that's
what
why
a
we do
virus does?
the gene sequencing. That's why we do cell culture. It's a virus on it, or you can take the virus out. Well, they did some human exposure studies too, and even animals infecting them too. So you can that makes it what a virus, unless you have an. In other words, you can infect other animals with it. You can infect humans with it that've been exposed to it. So,
Okay.
I mean, it's pretty easy to. I haven't seen anybody. I mean, I'm not sure what you mean by a particle. It has to be infectious. You mean, is it replicating? If it's replicating, then it's a living agent. I mean, and it's a virus or a prion or some other thing. So, 'cause you
Okay.
can't possibly
Yeah. I
use
don't, what I'm saying is, um, in order for it to be determined that it's a virus, it needs to basically show that it causes illness. You need to somehow prove that it causes illness,
Well,
right?
yeah, not necessarily. It can be asymptomatic. So you can be infected, you can be screening it, but you're not clinically ill. I remember it was
Yeah,
time
that part threw me for a loop. But but then that to me that's not very scientific. The the way that you can call something a virus from my estimation is that it's a particle that can cause someone to get sick, right? That was, every time we've had an epidemic, it's been this particle is going around making people sick. So in order to say that like that particle exists or that particle exists and causes illness,
right.
there needs to be some kind of evidence of that.
Yeah. Has anybody pointed you to Cox postulates? Uh,
Yes.
yeah,
Yeah, I've heard of them.
yeah. And then that's kind of the sort of thing you see if you isolate the virus, or you can
Hmm.
not even isolate it or give the virus from one person to another, like norovirus can't grow well in cell culture. We can take a stool and give it to another person and you can infect them from, if the person has the virus in his stool. Well, that's one of the things to do it. Is it a virus? Well, if it looks under the microscope like a virus and it replicates, if you can get it to replicate, then it's a virus. But I'm not sure. Uh, I mean, as far as COVID met all Cox postulates, so it's an infectious agent for certain. And all the data, nucleic acid data, uh, and the cell culture, I don't see why there would be any question.
Okay. How did COVID meet Cox postulates?
Oh, because you can you can transmit it from one person to another is the basic one of Cox postulates, and you could isolate the agent. You got to see it. You could identify it. You could give it to another person or animal, and they would get infected.
Okay. But you also said that there could be asymptomatic people.
Right? You could be asymptomatic. Yeah. That
but
means we
but
know
then
you're infected, and because you're excreting the virus in your feces, your saliva, your urine, that's how we know you're infected, and you developed antibodies against it.
so Cox postulates requires that um that it it the virus is isolated and then it's put into like a human subject, or transmitted to a human
Animal
subject,
or an animal.
animal, and then there's evidence that they're sick as a result.
Well, sick, or you can isolate the virus from their stool, feces, or they develop antibodies.
Well, to me, that doesn't really that's not how I've heard of Cox postulates. It's the purpose of Cox postulates is to prove it's a scientific experiment that proves that the agent is actually causing illness. And the only way that that can be proven is to run those experiments and then witness or observe the specimen or the patient, and then if they're sick, then that's evidence that something happened. But then you have to do it again multiple times. You have to do it with multiple people, multiple patients, multiple animals. The issue with this that you're talking about is that they could be asymptomatic and not show symptoms. And so how can you prove that this is actually a pathogenic particle if your cop out, so to speak, is, you know, it's you can just say they're asymptomatic.
Yeah, no, that's not a cop out because some viruses are almost always asymptomatic. Reovirus, never heard of it probably. Almost everybody gets it, but almost nobody develops an illness that we can identify. Rare illnesses. Now,
Okay.
and you can become chronically infected the rest of your life. Like with herpes, you get herpes, you got it the rest of your life. Now it may be asymptomatic, it's replicating into you, but unless you get stressed out, or let's take shingles. Shingles, right? That's when you get. My wife just got a case of. She had when she was a little kid. She had chicken pox. Now she looks terrible on the back. That's an asymptomatic virus that now is symptomatic. It won't happen with everybody, but the virus is again replicating and producing to a great extent to produce the illness. So asymptomatic doesn't mean you're not going to get ill at some point in the future either. It's just you're asymptomatic right now.
Okay, I understand, but okay, I can see what you mean when you say it's not a cop out,
Yeah.
but at the same time, it's not evident in the moment that you're running the experiment that that person is sick. And so it's hard to jump to any conclusions based on the fact that they just have a particle, that you basically gave them in their body, and as you say, it's replicating. Um, to me, that's not that could just be any particle. It doesn't have to be a virus. Um, what what I'm trying to understand is when you have the particle, um, and you run an experiment, how do you prove that that particle is making someone sick? The way that in my mind I would prove that is I would give them, the sample, and then they would get sick. If they didn't get sick, it could be asymptomatic, but in that moment, you can't jump to a conclusion saying that it's actually a pathogenic particle. It could create illness for some people and not create illness for other people, but that doesn't necessarily mean that it's a virus.
But since it created illness in some people, it is a virus. And by looking at an electron microscope, by the genetic material, by the antibody responses, Yeah, you can say it. I don't know of any self-replicating disease-causing particles that aren't viruses or other related things to it. There ain't
Okay.
nothing out there.
So as long as there's a few people that get sick from it, then it's a virus and
You
it's
know, it looks like it under a
replicating.
microscope. We have viruses we can find in people. We don't know what illnesses they cause. We know they're replicating in their body because they're always there. They're excreting them. But what do they cause? We don't know. There are unknowns. We can look at the genetic material, can look at the capsids, electron microscope. We see them. They're replicating into people, but as far as we can tell, they're not causing any illness. So that viruses can do that too. Viruses don't have to make you ill. They can just use
Okay, but
the growth medium.
certain ones, okay, certain ones that are, that are dangerous are the ones that,
We call it
make you.
acute infections versus chronic or infections that you call on it. So
Okay. Um, so I guess the trouble is it's in that um like the viruses, sometimes they cause illness, sometimes they don't. Um, so how can you really know that something is a virus or something, is causing illness or is dangerous potentially?
well, you have to do that, but epidemiological evidence to see one, if it's a dangerous, are people getting ill?
Hmm.
Then it's potentially are people dying from it. We're isolating the virus from their stools, their serum, their antibodies reacting. They have all the classical characteristics of an infection by a virus, and you can see it in the stool, in the blood, in the serum, wherever it is in the tissues of the person.
Hmm. Is there a chance that it's just the body going through some kind of detoxification?
Not if you're excreting the virus and you're ill with it, and you produce your antibody titers rising to the agent. That would seem to be good evidence that you're infected.
What do you mean?
Well, one, you have all the characteristics. You're excreting or secreting the virus. You can find the virus nucleic acids. You can see the capsids. You can see your antibody response going up as your body tries to fight off the infection.
Okay. So yeah, so you're just judging it based on like the body's reaction.
Body's reaction to your ability to isolate the virus, identify the nucleic acid in you. Um, you should be excreting the virus,
Okay.
or you'll have the virus somewhere in your body, in your blood. Your antibodies are responding to it. You're making antibodies specifically against the virus that's infecting you.
Okay. So going back to the how viruses are isolated by those who develop the tests.
Right.
Um, so in order for it to be an accurate study, you would need to have a healthy person's spit, or you need to have a sick person's spit in the cell culture process. That's what usually is done. Then you'd need to have a control experiment, which is with a healthy person's spit, also run in the same cell culture, the same type, not the same exact, you know what I mean? Two cell cultures made up of the same uh, ingredients, basically.
In a lot of clinical studies, they do that. You always have to watch out for asymptomatic people, though, because they could be excreting the virus and not be clinically ill.
Okay. So,
So a lot of
so
they did. Let's take human feeding experiments with virus. They do run controls where they give people placebos without the virus as a control them versus those people who may get the norovirus in human feeding studies to show, because they're trying to determine what level of virus will cause an infection. So they don't do those many studies anymore in humans because I'm trying to think of the right word for that. They don't allow those anymore on it. But usually, what we used to do for norovirus, which I guess it has been a while since they did those. They have people they gave placebos, and they gave people who they gave the virus to show that the virus infected and what level of virus was necessary to cause an infection. They used to run, but those two, but they've done those in human feed. They don't do it with every virus now because it's not considered ethical to do that kind of work now. They did a lot of that work actually in the 40s and 50s in the Army using conscientious objectors. Said, well, how would you like to be part of a salmonella study where we feed you salmonella? Because they could treat them with an antibiotic, and they waited until they became ill, and then they gave them an antibiotic right away. But those kind of studies are considered unethical today to do this.
Right. Okay. So, but going back to I don't know if you have experience outside of virology in like the science field, but I'm sure you seem like a smart guy. So you understand the importance of making sure that a study can be replicated, duplicated, and also that every part of the process is proving something.
Right.
So the issue that I have with the control experiment, or maybe the lack of control experiment, because I'm not sure how many of these control experiments are run, but the fact that so you're saying that if there's a healthy sample and it's run through the cell culture, that if it doesn't show particles like viruses, then then it's a healthy sample. If it does show particles, then it's an asymptomatic sample. Is that correct?
Could be, or it depends who you got the sample from.
Okay.
Person who's clinically ill, well, then I would say that might be the agent that's causing the illness.
Okay. What if you have someone? Okay. No, I'm not going to go there. So I guess the trouble that I have with that study is that, and if the sample creates the multiplication of the particles and it's a healthy sample, it could very well and that person, let's say that person has no symptoms at all, and they're not ill at all, and it still creates those particles. What would you say is happening there? You would say it's asymptomatic?
If the person is not clinically ill, that's a definition of an asymptomatic person. He could be excreting the virus. We could find lots of virus. Some people will actually excrete it all their lives and never become ill. I remember, who was the poor guy with a Giardia infection? He was chronically ill in Colorado, and he sold his stool samples because he was able to produce all the Giardia laboratories needed at one time. So you can become chronically ill for all your life and be excreting an agent for years and years.
Okay.
Particularly if you're somewhat immunocompromised. I remember some people they found became chronically ill with the vaccine strain of poliovirus. They were screening it all their life, but they never got ill with polio. So that can happen. But maybe,
Okay.
yeah.
So what happens in that control experiment if if the issue is what's happening inside of the cell culture
Mhm.
instead of um
An
like
animal,
just
human,
I'm sorry?
an animal or human, you mean? The virus will grow and reproduce, and it'll kill the cells usually.
Right. So
Ours.
some of the argument that the people have that I've learned from online is that they um the there's a chance that what's happening in the cell culture is a result of the ingredients in the cell culture being mixed with just, a fluid sample. And and that what's happening in there when the cell is multiplying or when the particles are multiplying is that it's basically like a reaction, like a chemical reaction that's happening in that cell culture.
That would be odd. I think they probably had too many margaritas. But the what we usually do is in the clinical samples, when I used to work with, we filter the sample to remove all the debris. So you're of a pore size of a filter that viruses can pass through to remove the bacteria for one thing, and other types of debris from it. So those are removed. Now, yeah, you can have what we call, because I work with environmental samples, you see more cytotoxicity, where the stuff in the sample is killing the cells. Uh, but usually you can dilute that out with it. But if you're good at this, we call it cytopathogenic effects. You can tell a virus from a chemical. Chemical ones are usually like that, and a day kills the cells. But you don't see
Hmm.
it with the viruses take days or weeks sometimes to grow on it. And you can see them increasing in number. It's just not, sure, you might have cytotoxicity, but you can also pass your sample, dilute it out if you want, because we run into cytotoxicity problems with chemicals in environmental samples. So we dilute it out, or we put it through a resin to remove the chemicals, or we diluting it out is a little bit easier. But those are issues that we deal with environmental samples all the time, but they're easily resolved by a lot of techniques and that. And then the other thing is what you can do is you can look under the electron microscopes or do your PCR to determine if the virus has been growing. So that the virus is growing. It doesn't matter if there's any chemicals that may be tied. The virus is growing in it. So those are pretty easy to deal with. As a environmental microbiologist, chemicals in the samples are always an issue, but we have a lot of ways to deal with it. And then we get a good idea whether the virus is CPE, and then we confirm it by doing molecular methods to detect the virus. So I don't I mean, I have more of a problem than any clinical person has with that, because I gotta deal with sewage, which has got about every damn chemical in the world with it. So we have to deal and bacteria and parasites. So we have to remove those before we do the virus assays and the chemicals too. So there are a
Okay.
lot of techniques to deal with that.
So so I guess going back to the to how you know that something is a virus versus some other particle is the way that you know it's a virus because it's replicating or and also because it looks like one.
And because I can sequence the nucleic acid and say it's a virus, you just have to grow it up, sequence it, makes it pretty easy to tell it's a virus, what type of virus,
Okay.
or a new type of virus.
So how do you know that virus is going to lead to sickness in it or illness in someone who is exposed to it?
Or you can do it by collecting clinical samples. That's part of the evidence. Also, no, there were human feeding experiments with the uh, coronaviruses that cause respiratory illness, right? Where they gave it to human volunteers years ago. Uh, so we do know the coronaviruses will replicate in human beings and cause disease because they did those studies.
Okay, so do you know what the last study was, or the
I
last
thought I read
group
that
of studies?
it goes back probably to the 1970s when they were doing that kind of work. I thought they also did it with the SARS-CoV-2 in England. I thought they were doing some studies on it. I thought I read too. I'd have to go look and see if they've been published. But yeah, human coronaviruses were done. The 229E was done in human volunteers, I think in the 1970s, to get an idea of the infectious dose.
Hmm. So when COVID happened, how did they know that this new agent, if they haven't done studies since then, how did they know that this new particle is going to, cause illness?
How do they know? Well, they isolated it from people who were clinically ill, and they saw it transmitted from one person to another at shared facilities. So, and then they looked at again antibodies, nucleic acid. You could look at the cell
Wait,
culture.
how did they see it transmitted from person to person?
Oh, by people sharing in the same room. If you've seen people riding on the same buses got the same agent. I've seen ones where they even sequence it to show that it was the same one infecting the other person in households.
Hmm.
They've done a lot of it
Okay.
in
So
shared.
it was more based on like, you know, uh yeah, they were just looking at like normal circumstances that people were in and showing that, people that were around each other were getting each other sick.
Right, that's right.
Okay. So,
Isolated from the air too. Isolated the virus from the air in some studies.
hmm. So how do they know that it was not like you know, I guess a typical flu season or, flu and cold season where people just happen to be getting sick around the same time?
Because the flu and colds went, the incidence of it disappeared almost because
Hmm.
COVID took over. You can, it's interesting to look at the epidemiological charts. The influenza just dove the number of clinical cases and common cold. And now, and then I don't know if you read the literature, but they did a huge surge when COVID decreased in numbers. We've had record. I mean, like norovirus is even a classic one. It almost disappeared because everybody wasn't going anywhere. They were staying home. They weren't eating out. They didn't get exposed to all these other viruses. So those levels went to record low levels. And now, when all the people stopped wearing masks, you could go back to school again. Then poof, the levels popped right back up again. So you can see the big decrease and big increase of
Hmm.
all these other viruses because COVID pushed everything out of the way. Everybody, COVID not only pushed it out of the way, but everybody was staying home. They weren't eating out, so they didn't get exposed to all these other viruses. It was quite interesting. That's been one of the things
You
dealing with more outbreaks of norovirus since the COVID because it really had decreased.
you don't think it was an issue with testing, meaning testing for
Oh
COVID
no, oh, no,
versus
they were
testing
testing.
for the flu?
Oh, they were testing for it too because they wanted to make sure did you have COVID or did you have the flu. So they were doing the same amount of testing. That was really was one of the things they noticed. A lot of people said, well, we should really pay more attention to hygiene because when we practice more increased disinfecting, wearing masks, we had a huge decrease in all these other respiratory and enteric illnesses.
Hmm.
Now they're coming back
Okay.
up now. So yeah, in fact, record levels in some places.
Okay. Um, so yeah, I would say uh I think we can wrap up this conversation. I'm not sure how much. Yeah,
Well,
if
I
you
got to
have
go on to
anything
another
else you want to
call
share.
here.
Okay.
But I'm willing to talk to you again if you want. It's no problem with me. I'm
Okay.
I'm doing it, but yeah, it's is it a chemical? That would be easy. There's a lot of parameters that would differentiate it because I have that issue with environmental samples. Is it a chemical causing cell destruction or is it a virus? So we have to run all these controls and test samples and that, but that can be dealt with. I have to deal with it all the time. with I don't think it's such a big problem in clinical, but environmental samples, that's a big issue. How do you know you really found the virus? So we have to do molecular techniques. We have to do multiple cell culture passes. We have to treat the samples to remove the chemicals. Man, yeah, more of a problem with my field than the clinical guys. They got it easy. Let me tell you. Feces is easy. You know, you got all these people dumping chemicals into the sewer, and I got to try to get the viruses out of that. So that makes it challenging. You know, so I think I'm going to have to run to my next call here. But if you want to talk more, just email me. I'd be happy to do it anytime.
Okay,
Sure.
awesome.
Good
Thanks
luck.
for chatting.
Okay, take care now.
All right, thank you. Bye.
Get this.